Okay, let's get started again. Hopefully, this is not getting to be too much of a broken record here. Cool seminars tomorrow. Yes, I know you probably think you should be studying, but these will probably be at least as important. Insights to the Evolution of Viruses and Antiviral Responses by Eugene Koonin at noon in room 247, SRTC. In the evening at the First Congregational Church, just down the park blocks from here, 7 p.m., free tickets available over in the box office. And then the chemistry seminar on Friday after the exam. And if your mind wasn't blown enough already, you can hear about building powerful new reactions between tetrazenes and trans-cyclooctene into proteins via genetic code expansion. Ideal bioorthogonal ligation within our grasp. So I'm going to go and try and figure out what this guy's talking about at that point. So yes, exam on Friday. As usual, Scantron, 50 questions, single best answer. You can have as long as you need on Friday to finish your exam. Any immediate questions on that right now before we do our clicker questions and half of you disappear? Okay, so without further ado, when translation stops, the presence of blank leads to nonsense-mediated decay. SR proteins bound to messenger RNA, exon junction complex bound to messenger RNA, exonucleases, translated poly A tails, or tmRNA. Ten, 5, so yes, it is the exon junction complexes. We'll talk more about SR proteins a little bit later on. So yes, we have B. Our second question. Protein folding chaperones, A, hydrolyze ATP, B, change their structures, C, block incorrect protein interactions, D, are involved in making viruses, E, all of the above. Ten, 5. Looks like nobody else is actually here. So basically, there seems to be two ideas. Let's actually show the results here. either C or everything. Those are our two options. And yes, it is more than C, which only gives you E as an option. Yes, they all hydrolyze ATP. When they hydrolyze ATP, their structures change. That has to do with how they're interacting with other proteins. And yes, of course, they block incorrect protein folding. That's what protein folding chaperones do in the first place. They're also involved in making viruses. That was the very first one of these that was found, is the GROE for growing the lambda bacteriophage. So E is correct. And any of you who have to leave, adios, good luck. We'll see you on Friday. And now I'm going to go over everything we've talked about so far relatively quickly. Again, please stop me as we go. This is all of the material since the last midterm. Yeah, wow, we were even talking about initiation after the last midterm. That seems like forever ago. So just a couple of important things about initiation and things that I think I probably did a bad job of talking about, discussing the first time that we went through them. Here, the really critical aspect of getting replication of your DNA is getting helix opening, getting those two strands apart. And so that is the critical part of all of the initiation processes that we've talked about. It's also critical if you think about transcriptional initiation. Same thing, got to get those two strands apart. Once that's happened in terms of DNA replication, you have primer formation. And important here is you have two replication forks. One fork that's going to go this way from your origin of replication, one fork that goes that way. And in each fork, you're going to have a leading strand and a lagging strand. Focus on the eukaryotic replication organs because there seemed to be some confusion about this. Yeah?
Oh, I think at one point you said a sentence like the critical thing is giving a helicase on. Is that similar or is this prior to that? Or?
We're getting to that in just a second here. So the critical case is it's pulling the two strands apart, which is the critical thing. And so putting the helicase on, you clearly have to do first. But in some cases, turns out in bacteria, just putting the helicase on is all right. In eukaryotes, the helicase is on and it actually doesn't start yet. So that's getting to this point. Where on eukaryotic origins of replication, you have your origin recognition complex, it binds to that double-stranded DNA at that particular position. You have these two cell cycle proteins, CDC6 and CDT1, that then help get the helicase onto the DNA. But this hasn't started yet. And so this is getting back to your question here about, when does that actually happen? What you have to have here, going from the so-called pre-replicative complex to actually getting those two strands apart and starting, is you have to get rid of CDC6, which happens through phosphorylation at the appropriate place in the cell cycle because of the cyclin-dependent kinases. Once that's removed, you also have the removal of CDT, and you can think of these as basically kind of brakes. They're held down, not allowing the helicase to move. As soon as the helicase moves and pulls apart those two strands, then you're going to get replication starting from that particular origin of replication. The phosphorylation of the origin recognition complex is a way to mark that particular origin of replication as having already been used. And so it can't associate with CDC6 and CDT1. It can't associate with a helicase. So you're not going to get replication from here until that phosphate gets taken off, which happens at the very end of S phase. And so that's what happens for these eukaryotic origins. The problem at the end of chromosomes for eukaryotic chromosomes, this is the figure that got eaten by my computer last time. It appears to be my computer, not the computer in here. And that is when you have a linear chromosome that gets replicated, it's easy to replicate the leading strand that goes all the way out to the end. However, since DNA polymerases can't start by themselves, they need RNA. The little green RNAs are here. Those then get removed by RNase H and or flap endonuclease here. And so you end up with a shortened lagging strand on one of the two copies of your chromosome. This, of course, will get separated in the next round of replication. And what will happen is the ends of your chromosomes will start to get shorter and shorter and shorter, and eventually end up taking up some of the important genes. How is this dealt with, of course, is through the activity of the telomerase. Again, the figure that got eaten by my computer last time. The telomerase, basically this piece here is just a blow up of this end right here. So this has a 3' OH and template strand where, again, the primer used to be. All that the telomerase does is it extends this 3' OH, giving you a longer and longer template, which DNA polymerase alpha, this was pointed out to me, will now replicate together with primase. You're still going to have this overlap at the end because for all DNA polymerases, they have to have RNA primers. So you're always going to have this little overlap at the end here. But this overlap is made of a repeated sequence because of the reverse transcriptase activity of the telomerase. It makes copies of the RNA that it carries with it. And so all these copies, and this copy here means that it's complementary to a sequence back here. So you can loop around and form these T-loop structures, which protects the end of the chromosome. Yeah?
All right, I'm confused where there's the RNA overlap versus where you have the DNA. So what exactly are the nucleotides that the telomerase is putting down versus then when the DNA pol alpha would take over? So I'm looking, at least on this slide, there's still the red included, and I'm trying, like the red nucleotides, along with the green. I don't know why it's not on that slide.
Okay, so the idea here, so I'll paraphrase your question, is what's being made by the telomerase and what is DNA versus RNA. So here, there is no RNA on here at all, except for the blue RNA, which is part of the telomerase. So here, this DNA polymerase would have extended from a primer over here. So the primers are missing on here. All of the red is DNA, which is made by the telomerase. Green is made by pol alpha. So there's no RNA on here.
So you just said telomerase makes both RNA and DNA?
No, I did not say that telomerase makes DNA and RNA. Telomerase makes DNA. Telomerase is a DNA polymerase that uses an RNA template.
Sorry. Apparently I'm colorblind. All right.
Usually males are more colorblind. My brother's colorblind, and this is, yeah, the red-green stuff here is a problem. He would think it's all brown. So sorry about that confusion. Most textbook people are supposed to take care of that. Okay, other questions on telomeres, sorry, before we move on. Okay, so DNA damage is something, yeah.
How long does the telomerase keep going, or how?
So the question is basically how many copies does the telomerase make? That's a really good question. Usually it's a couple of hundred copies, but it varies from organism to organism and cell to cell. So, but many copies. As far as we're concerned, many copies. Like my mathematicians friends like to say, one too many. Okay, so shifting gears a little bit, talking a little bit about DNA damage. Turns out my model got damaged, bringing it back from here one day. But that was a single-stranded break as opposed to DNA damage, which is happening at the bases. So most of the DNA damage we've talked about has to do with changes to the bases. Lots of different things happen, but it's mostly going to be due to hydrolysis. So here, hydrolysis is going to deaminate your cytosine, giving you uracil. Uracil gets chopped out by a uracil DNA glycosylase. Or you also have depurination, which is just hydrolysis of the glycosidic bond, which is between the sugar, which you can't see down here, and the base, leaving you with sugars without a base. And then, of course, 5-methylcytosine, you deaminate with thymidine, and this is a big problem that we'll talk more about later on. The only reaction that we've talked about that doesn't have to do with hydrolysis is what happens with UV radiation. which leads to these cyclopyrimidine dimers here. Could be thymidine, could be cytosine, any of these that get lined up here. So how does the cell deal with this kind of damage? Two different ways: base excision repair, nucleotide excision repair. This is the very precise one, but it depends on having a very specific chemical change. So you have to have an enzyme which recognizes the particular change. So here, for instance, the uracil, which gets cut out by uracil DNA glycosylase. You have an AP site here, apurinic, if it's just been purine which has been lost, so another way to get this DNA damage, or glycosylase, which has chopped something out. Once you have that, you have an endonuclease and an exonuclease that just cut out that one mistaken base, and that gets fixed by DNA polymerase and ligases. If, on the other hand, you have a big change in your DNA strand, this could be a pyrimidine dimer, could be lots of other things that we haven't talked about. But this is a very general technique where there's a recognition that there's a problem, but it's just a problem that has to be dealt with. Then there are two endonucleases that are going to cut on either side of that problem: helicase, which removes the problem, and DNA polymerase and ligase that hook this back together. So those are the two main processes for dealing with changes in bases. The other problem that we have, yeah?
When it cuts out a section of it, is there any rhyme or reason how big the section will be based on the formation that's in the DNA?
Oh, so the question is how big a piece is it cut out, basically? That has to do with the proteins that are recognizing it. So we talked about UVR A, B, and C. Those are proteins that will sit down on that one particular change, and then through the protein-protein interactions, it'll be that distance away from where that particular change is. The other thing you'll notice is that this is usually going to be on the order of 10 nucleotides. Why 10 nucleotides? It's one turn of the helix. This can be on the same side that those happen. Yeah?
What does the cell do with the junk base pairs, the junk DNA?
Oh, this piece, which has been cut out, that will get chopped up by our friends the exonucleases that love to chew on the ends of things into individual nucleotides, and then will get recycled. Yeah?
So with base excision repair, it's uracil DNA glycosylase just for the uracil. Are all of the things that will remove one of those not uracil B, whatever blank DNA glycosylase?
Correct, yes. So for instance, we talked really briefly about aatOxo-G, which is a very common oxidative damage that happens. There's an aatOxo-G DNA glycosylase. So for these very common kinds of changes, for very specific chemical changes, those will be recognized by a particular DNA glycosylase. Yeah, you're exactly correct. Okay, so that's what happens with bases. What about other things? And again, what happened to my poor DNA model? One of the DNA strands here got broken. So a single DNA strand break, in theory, you could have DNA ligase that would come in and religate those two ends together. However, you have replication happening, which happens particularly if you're thinking about bacteria, but also true in eukaryotic systems if you're in S phase. Replication fork with a leading strand here, that leading strand is going to continue all the way along here. The helicase is going to be pulling these two strands apart, and once it gets to where this single-stranded break is, if it hasn't been repaired, now you're going to end up with a double-stranded break here, because it's just that new DNA which has come along, gets to the end of the template. All of our DNA polymerases need templates, so it stops. And so we've got a new strand and the old strand that had the single-stranded break in it. That double-stranded break is probably the most common form of double-stranded break that you see in bacteria, but also probably also in eukaryotic cells. And how does that get dealt with? It gets dealt with in exactly the same way that our homologous recombination process we've talked about before. You end up chewing in at the 5' end. You have a 3' extension with an OH at the end. That can now base pair with the opposite strand down here. Now it's base paired. You can extend from this piece Now we've got a Holliday junction here that has to get cleaved and rejoined. Once that happens, you have a nice double strand here with a repaired piece. That's where this DNA nick was in the first place, and normal leading and lagging strands. So that's the vast majority of double-stranded breaks that occur in normal cell, normal replication are actually happening because of this. A single-stranded break and then replication through that, which leads to a double-stranded break. Yeah?
In the second picture down on the right, does
This one?
progression of the junction stop while it's trying to be repaired, or does it continue on?
I'm not sure I quite understand your question. Sorry.
The top double strands
Yeah.
are being connected with the other DNA. Will the replication fork that you see on the right continue on and it will just catch up later, or does it stop while it's trying to repair through the Holliday junction?
Oh, so basically this piece right here, what happens to that?
No.
Or you continue along here?
Does it keep splitting apart?
Oh, does the replication fork continue, basically, is the question, while this is happening?
Yes.
Yeah, so that's an excellent question. Does the replication fork continue? It turns out that there's a whole set of really fascinating reactions with all kinds of proteins with names that I don't want you to remember that stalls the replication fork until that gets taken care of. And if you're more interested in this, talk to Dr. Corsell in the biology department, because that's exactly what his lab does.
Out of curiosity, why doesn't the exon nucleus degrade the 3' end? Did you explain this already? Or?
Yeah, so the question is, why don't you get degradation at the 3' end? And it turns out that there are lots of proteins involved in these replication forks that will recognize those particular double-stranded break ends and basically protect the 3' end and allow the 5' end to get degraded. And in fact, you have the nucleases, part of that complex, which will do that. Yeah?
The DNA nick, is that usually a mistake by the topoisomerase somewhere down the line that just made it through a fork?
So the question here is about the nick. Where does the nick come from? And could it be a problem with topoisomerases? Yes, it could be, but usually topoisomerases are inverting their reaction, so it makes a nick but then religates it. So the protein would probably actually be bound there if there were still a nick there. Usually this is just hydrolysis that happens. Now, hydrolysis happens in phosphodiesters. It's unlikely that it's going to happen right on opposite sides of each other because it's a random occurrence, but it's certainly going to happen. Also, if you think about the lagging strand is going to have lots of nicks in it because every time you take out a primer, you have to religate at the end after you're done with that DNA polymerization. Okay, so that's what's going on with this, how you get most double-stranded breaks. We also have double-stranded breaks that form because of nasty radiation. Radiation therapy, for instance, actually just talking to one of my colleagues who's undergoing radiation therapy right now, because those double-stranded breaks end up causing mutations. But if you have other double-stranded breaks, and particularly those that form specifically during meiosis, those are double-stranded breaks that are made by endonucleases that are induced in meiosis. So you're specifically trying to make double-stranded breaks. And then once you have these double-stranded breaks that have two continuous strands, as opposed to just that one strand that you have in replication forks, then you have basically exactly the same thing that happens. You cleave in at the 5' end, the 3' end will go and invade an opposite strand, you end up extending these, end up with these Holliday junctions, and these Holliday junctions, which we'll look at in a little bit more detail on the next slide, then get cut and religated to each other. And that can generate two different kinds of chromosomes here. And so the idea here is that what we started out with, we had yellow at the top and red at the bottom. Sorry about the colors here. Yellow at the top, red at the bottom. You can have two different kinds of what they call resolution of your Holliday junctions, which is cutting and putting them back together. You can either end up with chromosomes which are the same at either end, so yellow at this end, yellow at this end, and red at this end and red at this end, or what is called the crossing over, and the crossing over you can literally see in meiosis is where the sister chromatids have crossed over with each other, where you have now the red information at one end, and continuing beyond where you had this change, you'll have the yellow information, and of course vice versa. And all this has to do with how these Holliday junctions are cut and religated to each other. And I abhor this particular image. I much prefer this one in terms of thinking about this. So this is that crossed over structure, which is supposed to represent one of these guys right here. That's not what Holliday junctions look like. What Holliday junctions really look like is like this, because they're double strands that are wrapped around each other and linked between the two different strands. You can also see that here, where we've got different strands that are exchanging with each other here. So it's a lot, I think, better to look at Holliday junctions like this, because this is really a much better indication of what these guys look like. So what I wanted to do here is literally draw on here what happens. So here, if you cut and ligate, you can either cut across here and ligate these two ends together and those two ends together, and that would clearly give you a crossover, because you've got now reds here and yellows over this end. If, on the other hand, you had a different colored pen and cut this way, now you ligate these two ends together and those two ends together. You don't have a crossover. You see it's red here and yellow here and yellow there. So it's just how you cut and religate is going to depend on whether you get a crossover or a non-crossover. Does that make more sense? Less sense? More confusion? Less confusion? Yes, kind of, sort of. Pardon? Yeah. And so this, I still have my pen on? No. Oops. Turn the pen on here. We're looking at, not an arrow, a pen. This guy here on our next slide. Right, so 50-50 chance of going, pen doesn't remember that I want to have a pen here. So 50-50 chance of going this way or going that way. So 50-50 chance one way, 50-50 chance the other way, as far as this class is concerned. Turns out there are other mechanisms, but yeah, we're not going to get into those. So the questions on recombination, et cetera, at that, from this point of view. So let's talk a little bit about transposons. There seemed to be some confusion about transposons, so I wanted to spend a little bit more time talking about them, particularly the DNA-only transposons, also called the cut and paste transposons. Here you have a transposon which has some short sequences. Everything, again, sorry about the colors here. They're red, which are these inverted repeat sequences, yellow in the middle, which is the rest of the transposon, which usually encodes the transposase gene, and another little red inverted repeat at the other end. Inverted repeat just means going from 5' to 3' on this strand, you'll have a sequence. The same sequence is going to be over here on the other strand, going from 5' to 3'. So these inverted repeat sequences are what the transposase protein binds to. So it binds to these inverted repeat sequences, and then will cut out the transposon between these two inverted repeat sequences, because that's where the transposase is binding. Then it will go and find another place to jump into. And what happens here is that transposase will have cut out the transposon here in a way that it's just the transposon with those inverted repeats at the end. But where it cuts into the chromosome that it's going into, where it's jumping into, it cuts slightly offset on the two different strands. And so with that offset cut, you're going to end up with a gap here shown in black that then gets filled in by the normal DNA polymerase. Same thing is going to be true over here on the other side. This sequence between the two here is now going to get copied because it's been pulled apart, two different strands that will get copied by the normal DNA polymerases. That generates a direct repeat because it's now the same sequence on the same strand. It's just taken this sequence and copied it, the sequence between these two arrows here. Does that make sense how we get now these direct repeats outside of the actual transposon? Okay, good. So those are what you have quite commonly in bacteria. These are the DNA-only transposons that are cut and paste. The most common kinds of retroelements or transposons we have in our genome are the non-retroviral retrotransposons or retroelements. This is what they look like in the genome. It's a stretch of DNA that encodes usually two different proteins, a reverse transcriptase and an endonuclease, followed by a whole stretch of A's on one strand and, of course, T's on the opposite strand because this is double-stranded DNA. This gets transcribed by the normal RNA polymerase II, gives you a RNA with a poly-A tail, processing, everything is normal. Then this gets translated into reverse transcriptase and endonuclease. This now will bind to and cut double-stranded DNA. That's what endonucleases do. It cuts where you have usually a T, which will base pair with the A's in the poly-A tail that you have at the end of this transcribed retrotransposon. And then the reverse transcriptase will copy that RNA that was made and insert a copy now of this retrotransposon somewhere else in the genome. And one of the important things about this is it's now copying this. It's not cutting and pasting like we have with the DNA-only transposons. This is going through an RNA intermediate. This original retrotransposon is where it was originally in the genome. This one's going in somewhere else. So it's not terribly surprising that we have many, many copies of these, because over millions to billions of years of evolution, these have made many, many copies of themselves. And so it's a copying process. So 40-odd% of our genome is made up of these things. Yeah?
Is there a particular time when it's supposed to do this, or is it more random?
So the question here is basically having to do with timing. Is, I'll paraphrase your question. How often does this happen? Does it happen at a particular time in the cell cycle, particular time in development, et cetera? And the answer to that is it turns out that most of these retrotransposons are suppressed. And so under most conditions, very few of these are actually active in our genome. And there are a few cases of that. Seems to be happening mostly in early development. And as I may have mentioned in the lecture before, many of the cases of spontaneous hemophilia actually have to do with this. And it turns out this particular one, there are 4 or 5 different ones, but the L1 element moving into one of the blood clotting genes. Yeah?
When you say spontaneous there, you mean non-genetic hemophilia?
So, yeah, so the so question here is, that's fine. So non-genetic hemophilia, so it'll be a spontaneous change genetically which happens, hemophilia. So non-hemophilic parents, non-carriers will then have a hemophilic child who can then pass that on. And so it's that stage between the two parents which don't have that particular copy in one particular place. They have to have this gene somewhere because it's got to come from somewhere. But it's just in one place, and it's the copy and insertion in somewhere else which has caused the hemophilia in the child. And that particular case can, of course, be passed on by that child, should they have offspring. Yeah?
To put it in expectation terms, is it possible to find gene 0? Like, where was the first L1 element?
So the question here is, basically, where do they come from? And without a time machine, we're not going to know the answer to that question. You can reassemble some of these things, and actually, in fact, Dr. Koonin may be talking about this tomorrow. But if you line up all of these things and try and figure out a phylogenetic tree where they came from originally, you can get some ideas. But again, it's really not clear where these originally came from. My personal biased opinion is that they came from viruses exactly in the first place. But people also argue that the viruses may have arose from these as well. And there's probably some trade-off between the two. So, yeah, question that we really do not know the answer to. Yeah, sure. So what's the function of the reverse transcriptase? The function of reverse transcriptase is just like we had with the telomerase. So all it does is it takes a RNA template and makes a DNA copy.
And that's the first step, right?
Yeah, and that's this first step. As with all of the reverse transcriptases, well, actually, the reverse transcriptases that we talked about for the viral reverse transcription, it also can take DNA and make a DNA copy of that as well. So the reverse transcription is not just making this first strand, it also makes the second strand. And sign up for virology for next term, we'll talk a lot more about it, how that happens. Okay, so let's shift away from DNA and start talking about RNA. Basic message here is transcription. They're different parts of the genome, so you have to identify where you're going to start. And transcription is really nice because you can make many copies of RNA from one particular piece of DNA. So you can end up with lots of copies of one particular part of the genome. And then, of course, we talked about later, you can do translation and then get many proteins from any one of these individual RNAs. What I wanted to emphasize here is if you have a functional RNA, like, say, a ribosome RNA, then you have to have just this step, the transcription step, and for all the ribosomal RNAs, just one gene isn't enough, so these are multiple copies of these genes in order to get enough of these functional RNAs. How does transcription work? It's all about finding that right place to start. Once you've found the right place to start, then you've got to pull the two strands apart. So just like you have in replication, it's getting those two strands apart, which is really critical. Once you've started these two strands, one of the differences that you have between transcription and replication is in transcription, most cases you'll have this abortive initiation step where you just make a few nucleotides, spit them back out, make a few nucleotides and spit them back out. And the main reason for this seems to be that once you have transition from abortive initiation to elongation, this is an incredibly processive complex. And so it's going to make your RNA all the way until it gets to the terminator. And sometimes these are extremely long. Some transcripts are up to 1 million nucleotides long. We've actually seen a few of these. So they're ridiculously long. To make a ridiculously long transcript like that, you need to hydrolyze a lot of nucleotide triphosphates. So it's an energetically really major process, takes a lot of energy to do this. And of course, once you've made the RNA, that's then gets translated into proteins, and proteins takes even more energy. So it's this step. You really want to make sure that whatever gene it is that you're going to be transcribing, you really want to make sure that it gets transcribed. And so that's the abortive initiation step here. Yeah?
The book talks about abortive initiation as being a way to dislodge the sigma factor due to a strong bond.
Okay, so here, yeah. The book talks about sigma factor dissociation here and abortive initiation. It seems that the sigma factor dissociation here has to do with the transition from abortive initiation to elongation. We didn't talk about it, so it's not going to be something that's on the exam. But that does seem to be, again, it's a change in structure which happens going from your abortive initiation to elongation. And it's that change in structure which makes highly processive your polymerase. In the case of the bacteria, you lose the sigma factor as well. And in the case of eukaryotic systems, that's where you leave all those general transcription factors back at the promoter.
So is abortive initiation more often something that's kind of on purpose, like some signal says we didn't want this, or is it an accident, like it just doesn't get started?
So to some extent, I'll paraphrase your question, is why have abortive initiation? And it seems to be this happens at almost all promoters. You'll have abortive initiation. And so it's probably part of this step, the regulation between getting started and moving forward. And so that process, and in some cases you'll have more abortive initiation at some promoters, sometimes you'll have less at different promoters. But it also gives you a place where you can regulate how much of the RNA is actually being made. And we'll talk more about that when we talk about regulation a little bit later on. So another thing that I wanted to mention here, now this is specific to bacteria, and this is the bacterial transcription process. Everything else is the same here. Happens in eukaryotic as it does in bacterial transcription. The difference here is our sigma factor. The sigma factor is what allows the RNA polymerase to find the start where it's actually going to bind to the DNA in order to start transcription. So what's that called? It's called the promoter. Just wanted to emphasize here that this is just one strand of double-stranded DNA. So we're looking at DNA here. So from 5' to 3', TTG, ACA, TAT, AAT. On the other strand, it'll be ATT, ATA, etc. So double-stranded DNA. This is what the sigma factor together with the RNA polymerase is going to bind to in order to get the polymerase to the right place where it can actually start. And how we know about these is by lining up lots of sequences, finding how often particular nucleotides are at any of these sequences, and that generates our consensus sequence, which is also shown down here in the logo at the bottom. So just a consensus sequence is the most common nucleotide at any of these individual positions. So promoters in bacteria are going to have these -10, -35 sequences. In eukaryotes, they have the Tata box and the BRE, which should be labeled here as right next to the Tata box. The Tata box is, again, bound by the incredibly creatively named Tata box binding protein. TBP, which is part of the TF2D complex. Then you have TF2B binding, and then lots of other factors happen, but I haven't talked about them too much, so don't worry about them for the exam. And then you have TF2H, which associates with this whole complex. TF2H has the helicase activity for pulling the two strands of DNA apart, allowing you to get abortive initiation to start with. And then the phosphorylation of the C-terminal domain of RNA polymerase II with the TF2H kinase activity. So TF2H does two of these things. TBP binds to your Tata box. TF2B binds to the BRE. Once you have this binding, the polymerase can bind. Then you have TF2H that has the helicase and kinase activity. Those are the important ones as far as we're concerned. A whole bunch of other things, different transcription factors which are there too, but we're not going to talk about them. Yeah. Oh, one of you, sorry. Yeah.
What's your CA25 box there?
Oh, CA25? So approximately at -25 relative to the transcriptional start. And that's one of the things which is also different with bacterial versus eukaryotic transcription. The Tata box can be actually quite variable, as opposed to -10, -35, which is what you see in the bacterial promoters, which are always at those positions relative to the start site. Yeah?
What exactly does the phosphorylation do? Is it like what allows it to start transcription instead of abortive initiation?
So what does phosphorylation do here? What does the activity of that kinase do? It does two things. The first is it helps with escaping from abortive initiation. The second thing it does is provides a landing pad for all of the modifying proteins that are involved in modifying your messenger RNA, the capping enzymes, the spliceosome, the tailing enzymes. So doing those two things. There's a question up in the back there, I think. Yeah.
I can't remember you mentioning a couple lectures ago about how TF2H does not use quite as much ATP as a normal helicase would, or it's not sort of second to ATP.
Right. So the difference here is between TF2H and what you think about in terms of the replicative helicase. So the replicative helicase is hydrolyzing ATP all the time because it's always pulling apart the two strands. Once you've started with getting these 2 strands apart here, you don't need TF2H anymore because the polymerase has that isomerization. It sticks that little piece of protein in between the 2 DNA strands. So it doesn't need any more helicase activity. Whereas in the case of replication, that helicase has to be pulling those 2 strands apart the whole time. The question's here. Yeah? The BRE is the TF2B recognition element. So again, elements are always something which is in DNA, and factors are always going to be proteins. Yeah?
What's TF2A doing specifically?
Don't worry about it. We can talk about it offline. It's really interesting, fascinating stuff, but yeah, not important for Friday, which is after all that we care about. So, so once we've made the messenger RNA for bacteria, it's easy because that messenger RNA gets bound to by ribosomes. You make the protein. Great, efficient, wonderful. On the other hand, something which I think I didn't do a good job of explaining previously is that when you have transcription that takes place on eukaryotic DNA, you end up with what they call here a primary RNA transcript. Also, many people will call this the pre-mRNA. So this contains all of the exons in blue here and all of the introns in yellow. So people are not going to talk about this as being a messenger RNA. It's only really a messenger RNA after you have all the processing that takes place, after you put the cap on it, after you do the splicing, after you put in all of the poly-A tails. And so it's, people talk about, pre-mRNA processing or processing of this primary transcript. So what is that processing? Of course, it's capping, splicing, and tailing in the process. We'll talk about those again a little bit more here. So the cap is this 5' methyl G cap that binds 5' to 5' at the end and is really blocking the exonucleases, which otherwise love to bind into the end. Also provides the site for binding for, again, the creatively named cap binding complex, which helps you get that messenger RNA out of the nucleus. That then also will bind to, the cap will bind to the initiation proteins for the eukaryotic translation machinery. Yeah?
So your image is titled Eukaryotic mRNA and your slide is titled Eukaryotic mRNA?
Yeah. So this is concentrated on the eukaryotic mRNAs, the cap exons and tail, because the prokaryotic mRNAs do not have cap exons or tails. So it's, I'm concentrating on the eukaryotic messenger RNAs here.
Oh, okay. I'm on board now. I didn't realize it was both.
Yeah, they're both here. Okay, so let's talk a little bit about splicing. Again, there seems to be some confusion as far as splicing is concerned. There are really 2 different kinds of SNRNPs here. So small nuclear ribonucleoprotein complexes. There's U1 and U2. U1 and U2 are really providing the specificity. Where are you going to splice? U4, U5, U6 are actually doing the job of splicing. So if you have appropriate association of U1 and U2, then U4, U5, U6 will come in and perform the splicing reaction. And how exactly that works, again, fascinating process beyond the scope of this course. What happens? Binding again, U1, U2 gets helped by the activation factor and the branch binding protein, will bind to this A in the middle of your lariat. That's where you have your 2', 5' bond that forms, again, catalyzed by U4, U5, U6. And then this OH can attack the 5' phosphate over here on this exon, then leading to your splice product. This, in and of itself, cutting phosphodiester bonds, making phosphodiester bonds, cutting phosphodiester bonds, making phosphodiester bonds, there's no energetic change that happens there. The energy that you need for splicing has to do with changing base pairing interactions. And that was what, I think, again, I did a horrible job of explaining here. So To start with, you've got U1 that associates here at the 5' end of your intron, but in order to get splicing to take place, you actually need to have U6 bound to the 5' end of your intron. And that change, going from one to the other, that requires ATP hydrolysis. Because what you're doing is you're pulling one pair of RNAs apart and putting another pair of RNAs together. And so that's what all of the energy that you need for splicing does, is it pulls strands apart, allows them to come back together, pulls apart, puts them back together. So the actual reaction itself doesn't require any energy because it's breaking and forming the same bonds. But pulling apart these hydrogen bonds and putting back other ones, that's where you need ATP hydrolysis. Another thing that I think I did a crummy job of explaining the first time around is, to some extent, how does the cell know what's an exon and what's an intron? One of the ways is talking about SR proteins. SR proteins bind to exons. They bind only to exons. They don't bind to introns. So if you have SR proteins bound to your pre-messenger RNA, that says, hey, this is a place that we want to splice. Another way that the cell seems to know that these are particular places to splice is that exons are almost always the same size. They're about 150 nucleotides in length. And so the splicing machinery seems to be designed to, or evolve to, not intelligently, evolve to just take these 150 nucleotide pieces and hook those together. So it's the size of the exons and the presence of these SR proteins that really leads to getting proper splicing to take place. So we've done capping, we've done splicing. What's left? Putting the tail on. Here again, getting back to your question about why do you have phosphorylation by the TF2H on the CTD? That's because you have binding of not just the tailing proteins, but also the capping and splicing proteins, all here to the CTD. So they're right next to the messenger RNA as it's being made. As the messenger RNA is being made, there'll be a specific sequence in there. The sequence itself is not important for this class. Binding by your cleavage and polyadenylation specificity factor, then binding to a separate site by the cleavage stimulation factor, the endonuclease, which will cut in this pre-messenger RNA, because it's not done yet, we haven't put everything on it, cuts there, gives you a 3'OH that gets extended by the poly-A polymerase. The poly-A polymerase then makes a whole bunch of A's. Again, getting back to a question that I'm trying to preempt here, how does it know how many to make? It's around 200, but it varies a whole bunch from different RNA to different RNA. It seems to be pretty stochastic. And then poly-A binding protein binds to that, and now we have our mature messenger RNA that gets exported out of the nucleus and ends up getting translated. So now we have our messenger RNAs. All happy with messenger RNAs? Yes? Good? Happy? No? Unhappy? Questions? Yeah.
Where does that cut happen?
Pardon?
Where does that cut happen on the pre-messenger RNA?
When does the cut happen?
Yeah.
Oh, so it happens as this sequence is being transcribed by the RNA polymerase. So that sequence is transcribed. The CSTF and CPSF are going to bind to that RNA as it's being made, and cleavage will happen. So it's a co-transcriptional process. As that RNA is being made, it will be cut, and the poly-A tail will be formed. Okay, I wanted to, again, talk briefly about some of the other SN RNAs, only these are the SNO RNAs, small nucleolar RNAs, as opposed to the small nuclear RNAs. The important thing here really has to do with what these RNAs are doing. And I like to think of these RNAs, the SNO RNAs, as being very similar to what happens with U1 and U2 in splicing. They're providing specificity. They're through base pairing interactions, identifying a particular place on your RNA to do something to it. In the case of the snow RNAs, these are binding to the ribosomal RNA at a specific sequence, leading to modification that happens. So here, modification is pseudouridine. Here, modification is O-methylribose. A number of people ask me, what do I mean by these box CD and box HACA? Box CD just means a conserved sequence which is present in all of these different snow RNAs. Why are there a whole bunch of different snow RNAs? I say about 200 of these. It's because the sequences that they bind to here are all slightly different. And so each of the modifications that you're going to have in the ribosomal RNA is going to be dependent on base pairing by one of these small antisense RNAs. These small antisense snow RNAs also have conserved sequences which are going to bind to the proteins that are important for making pseudouridine, protein that's important for O-methylribose formation. Yeah?
Can you just clarify these terms, sense and antisense? Because it seems like they're used sort of everywhere.
So sense and antisense. Sense just means coding in terms of your protein. So it's going to be the strand that has AUG on it. And if you've got a antisense, that's going to base pair with that in the opposite orientation. So that would be your antisense. Now, that's great and wonderful. I'm sure somebody here is going to say, but Dr. Stedman, this is ribosomal RNA. It doesn't have proteins that it's coding for. And so this is now the RNA which is being made, then the major RNA, in this case, the ribosomal RNA, now the antisense to that. The sense will be the ribosomal RNA, what's being used. The antisense is then going to be complementary to that. So, yeah, again, and it always has to be opposite strands. If you think about codon-anticodon interactions, that's also sense. Sense is going to be your codon. Anticodon is going to be antisense, because it'll be the opposite orientation.
So the sense strand is always sort of the template strand.
The sense strand will be what has actually been made by your RNA polymerase in this particular case, like your messenger RNA. That's your sense strand. That's what gets translated. In the case of these ribosomal RNAs, it's what get used in the ribosome. Again, it's always going to be a definition because these RNAs have to come from somewhere. They've clearly been made too. Just briefly talking about the genetic code. Here, the main message is that the amino acids with similar chemistries group really closely together in the genetic code. And so anything which is brown here is going to have very similar chemistry, dark brown, green. This is actually any anomalous trichromats here, and probably the males. Are impossible to tell the difference between these two colors here, this brown and this green. But at least the red should be a little bit more obvious versus the blue. But again, all of these proteins that have, or say, amino acids that have similar chemistry are going to be close together in genetic code, which means that any kind of nucleotide change is not going to make a big difference in terms of your protein. So that's the main take-home message here. And ooh, interesting changes here. Amino-acyl tRNA synthetases, this is what decodes the genetic code. Important here is that we've got one amino-acyl tRNA synthetase basically for every amino acid. What that means is that these amino-acyl tRNA synthetases are going to be binding to multiple different tRNAs, because one is not, 20 different tRNAs. You've got up to 61 different tRNAs because they're going to bind to each of the different codons that you have there. It's less than that because of the wobble in the genetic code. But it's the amino-acyl tRNA synthetase interaction with that amino acid which is the most important. Here, the interaction of the amino-acyl tRNA synthetase with the tRNA, which is here in purple, this can now interact with multiple different tRNAs, multiple different anticodons down here, but it's always going to have one amino acid which it puts on the top here. Okay, so once you've got our amino-acyl tRNA synthetases, made your tRNAs with the correct amino acid at the end of them, now you have a ribosomal cycle. Here we don't need helicases, unlike replication and transcription, because it's just one molecule here. You have to have association of the small ribosomal subunit with your start codon and the initiator tRNA. This is different in bacteria and eukarya. Hopefully we'll talk about it in just a second, but here this is a very general case. Then you have the large ribosomal subunit that comes down, and basically it's more like a clamshell between the small and the large ribosomal subunits. Once that clamshell is closed, then you can have extension of your amino acid chain. But it's getting the initiator tRNA into the middle, which of course is the P site, the transpeptidation site of the ribosome, has to happen before you have the large ribosomal subunit that comes in to this process. In bacteria, you have three main initiation factors for translation. IF2 is the really important one, binds to GTP. Once you have appropriate interactions of the initiator tRNA with the start codon here, then you have GTP hydrolysis, releases this initiator tRNA in the P site. These guys dissociate, IF3 and IF1, which allows you to get the full ribosome formation here. This happens in terms of getting the small subunit of the ribosome to the right place on the RNA. It's the Shine-Dalgarno sequence that we have in bacteria, and this is the RNA which is coming out of the RNA polymerase. So as soon as that particular RNA sequence comes out of the RNA polymerase, the small subunit of the ribosome will bind on. Then you have the initiator tRNA, which will get extended. Eukaryotes have a problem, and that is that your messenger RNA is being made in one place, and you need to translate it in another place, nucleus in the cytoplasm. So the same problem, getting the small subunit and initiator RNA to the start codon, has to happen. But how that happens has to do with cap binding. And once you have cap binding, the movement of the ribosome along your messenger RNA until it gets to the AUG. Once it gets to the AUG, your initiator tRNA and the equivalent of IF2, EAF2, GTP hydrolysis, that protein is removed. Now you get the large subunit that comes down. That large subunit has the transpeptidation activity, which will allow you to hook up your growing polynucleotide chain to the tRNA, which is in the P site. And so when you have an extending polypeptide chain, it's always bound to a tRNA, because the transpeptidation happens here from your activated amino acid, which is hooked up to the end of that tRNA. Translocation, how the ribosome moves, depends on the activity of other GTPases. The binding here in the A site also depends on GTPases. When you get to the end of translation, you end up with a stop codon. There's no tRNA that can bind to this unless you've got cool selenocysteine and recoding, which I'm sure we'll hear about on Friday in the chemistry seminar. But then binding here in the A site and eventual processing, which then allows the whole ribosome to fall apart, also needs GTP hydrolysis. And then your RNA is free to be retranslated by a small subunit and a large subunit that come together. They do this in a polysome. So you've got eukaryotic RNAs that have ribosomes that are running around them, following each other's tails. Once they dissociate here, they can reassociate here and keep going. Talk briefly about selenocysteine. The main thing here is that it's using a stop codon and has to have an extra secondary structure associated with it in order to get the tRNA to be bound and to have this transpeptidation reaction to take place. Just want to talk really quickly in the last minute about the 26S proteasome. The 26S proteasome has the protease activity in the middle here, but getting to the protease, that's the important thing. That's what these regulatory subunits out here are important for. They bind to polyubiquitinated proteins, but also, and as I think I forgot to mention last time, any exposed hydrophobic piece of a protein in the cytoplasm will be bound by one of these regulatory subunits and fed in here. So it's not just polyubiquitinated proteins, but also any unfolded proteins that will get put in here. We'll stop here. You got questions about any of the rest of it, hang out. I'll be here probably till about 1 o'clock.